Thank you. So again, as Philippe said, the aim of our work is to really better understand the progression of Shigella infection by applying really simple quantitative bioimage analysis techniques within an in vivo model of shigellosis. So I just want to take a step back to really the beginning of microbiology, back into the laboratory of really the father of microbiology, Louis Pasteur. And this is during the late 1800s. And here we see an essential tool for all of us biologists, and of course it's highlighted in the talks today, and that is the microscope. So this is really a tool that's any microbiologist's best friend. And from the very beginning of this field of microbiology, the microscope has really played an integral role to the researcher to allow us the necessary observations to examine microbes on their scale. But unlike during the days of Pasteur, when physically drawing an image was the mode of capture, we can now capture images digitally. So these acquired images are in fact multi-dimensional data sets, so we have to look at them differently and think about them differently now. And they really harbor statistically relevant data. And the task now is to computationally extract that information. And we can apply these techniques to better decipher the complex interplays between host and pathogen, and moving beyond mere observations of times past. So in the end, to properly carry out quantitative imaging, it is really a simple combination of these three key ingredients. You need a relevant biological model, and in our case, it's one that's applied to host-pathogen interactions in Shigella. And then proper sample preparation and acquisition of images that really address directly the biological question at hand. And finally, there's the proper data handling and processing, and really trying to minimize and streamline any manual input to really keep data extraction as automated as possible. And only when all these components are taken into account can relevant quantitative information then be extracted, and therefore potentially reveal novel phenotypes as well. So we aimed in our study to apply these techniques, these quantitative imaging techniques, to examine Shigella within its host environment, which is the colon, the large intestine. So we begin with our biological model, and that is Shigella flexneri. So this is a gram-negative enteroinvasive pathogen, bacterium, and it uses a type 3 secretion apparatus. So it's like this needle-like structure that it uses to inject bacterial virulence effectors into host cells. And this induces invasion and cell-to-cell spread and the ensuing infection. So again, Shigella's main target for establishing infection is the large intestine. And the colon's main morphological feature in this tissue is the crypt, which are basically invaginations of the tissue. And at their base, they harbor the epithelial stem cells of the colonic epithelium. So now, during acute shigellosis in humans, infection results in a really acute inflammation, infiltration, and really a massive tissue destruction localized at the site of infection. So in our lab, we're actually using a recently developed guinea pig infection model to look at Shigella in its host environment, in the large intestine. So after interrectal injection of Shigella into the guinea pig, it results in a localized infection that's really indistinguishable from human shigellosis. So this is our model. And now to discuss our sample preparation and, of course, our choice of imaging system. And as we need to really maximize the amount of colonic tissue that we're imaging, so we're properly sampling the infection progress. So essentially what we did was prepare the tissue sample, or the colonic samples as a Swiss roll. So we take the 10-centimeter distal part of the colon, and we essentially roll it up and do transversal cuts. So this is 10 centimeters in length. So in the end, we have a massive amount of tissue that we really need to sample, and at high resolution so we can resolve individual bacterium and find their locations. So in the end, we had access to the Cell Voyager system, which is a spinning disk confocal. So that allowed us for this high-resolution 3D localization of the tissue and bacteria. It had a really sensitive camera, so we could image deep within the tissue. In fact, our tissue sections were rather thick. They were 30-micron thick sections. And then we could also tile the images, so we could look at a larger region at high resolution. And then finally, it was fast, which was great, because it cut down on acquisition time. So essentially, we built up this acquisition strategy where we would scan the entire Swiss roll of each animal we harvested at varying time points. In the end, I didn't mention it before, we harvested tissues at 2, 4, 6, and 8 hours post-infection in a total of 31 guinea pigs. So for each guinea pig, we had this large, expansive roll that we would scan at low magnification in bright field just to see where the tissue was. And then from there, we had to find the Shigella. And in our case, we're using GFP-expressing Shigella. So we used their GFP fluorescence to actually localize the individual infection foci along this full 10-centimeter length. And then for each of those positions, we went in and did a high-resolution 3D scan of every single infection foci along that 10-centimeter length. So in the end, this is just an example of an image that we acquired at a late time point. This is 8 hours post-challenge. So just to orient you a bit, we really did a simple staining. Just for the tissue morphology, we'll focus on actin staining and DAPI, so actin microfilaments and DAPI staining for nuclei. And we see here the muscle layer at the bottom, and then you can see the colonic mucosa, and this is a luminal side, and all the infection foci along this length of tissue. And in the end, this is a particularly large tile scan. So it's a large foci, so this is a 4x5 tile scan in high resolution. So if you zoom in the image here, you can actually resolve each individual Shigella. So then, once we have this large set of data, a large set of images, how do we process and handle this data properly? So in our case, we mainly used Fiji, which is just image J, the Amero database, and MATLAB scripting to extract the data. So this is just to step back and look at the numbers, the extent of this analysis. So in the end, we had a total of 31 guinea pigs. And from each of those pigs, we had in total, we had 172 acquired images from each Swiss roll. So we basically imaged all these Shigella along the entire length of each one, resulting in 172 images. And this took approximately 60 hours of acquisition time. But in the end, we were able to count tens of thousands of individual bacterium over a very large colonic tissue volume. And also to note, we had three different conditions here where we were comparing the invasive strain to a non-invasive strain. So our non-invasive strain actually doesn't assemble that type 3 needle, so it can't actively invade cells. So we use this as a comparison for the invasive strain. So with this large amount of data, we needed to centralize, and then to do so, we took advantage of the Amero databank. So this database basically allowed us to annotate, curate, and analyze our data, and multiple users could come in and access the data as well. So essentially, the raw data was all stored in this database, and then we had to carry out annotations as well, both manual and automatic segmentations. So first, with the manual annotations, we had to delineate the surface, basically define the mucosa, and then those main morphological features of the colon, the crypts. And we did that all by hand, by drawing in Roy's in Fiji. And then we did an automatic segmentation of individual bacterium using the plug-in TrackMate in Fiji as well. So just to give you an idea of what these annotations look like, we have our manual annotation, so we delineate the surface, which essentially we extrapolated across undamaged tissue surface. So you see an infection foci here and damaged tissue, but we projected the surface line across the uninfected, untouched tissue. And then the base of the mucosa, defining the mucosa, and then we have our crypt mouths, which are basically the opening of crypts at the epithelial surface, and then also mark their base, like where the stem cells are harbored. And then finally, we have the automatic annotations, which carried out, again, in TrackMate, so we can basically find all the X, Y, Z positions of each individual bacterium. So together, we can use this information then to pull out this quantitative data. But of course, we're dealing with images. So they're giving us an impression of the progression of Shigella infection. So they've given us an idea of what we should look at and what elements we should measure. So in the end, how is Shigella moving within the tissue as the infection progresses? So for example, what are the distances of each individual bacterium from the epithelial surface? And what's the dynamic of this penetration? And what is the resulting damage to the tissue? So again, just looking at our images, we have an impression of key events that are occurring. So this is just an example at 2 hours post-challenge. You see the Shigella here that are really closely associated with this crypt mouth. So this is basically the beginning of this invagination of the crypt. So we saw this occurring quite regularly at this early time point. And then as the infection progresses, we see that Shigella is already replicating, and that there's a bit of tissue damage that's occurring localized at the site of infection. So the closer you are to the bacteria, the more damage to the tissue. And this only progresses as time goes on. So this is, again, at 6 hours post-challenge. You already have a large amount of tissue that's being destroyed. And again, this is making, at later time points, it's very difficult to delineate morphological features at this point that are really close to those infection foci. And then at 8 hours, it's really exacerbated because now you have this damage that's being contributed to by both the bacteria and the host response. So again, there's this inherent qualitative element because we're dealing with images, we're taking pictures. But of course, now we can go one step further and take advantage of our tissue annotations that we drew, and then our bacterial positions as well, and then measure those dynamics, those interactions. So just to start, Shigella were designated as either tissue-associated or luminal based on their orientation from this epithelial surface that we had delineated. So accordingly, we could calculate a ratio, which is what's presented here. We calculated a ratio of luminal Shigella over the total bacterial population per condition per time point. So essentially what we saw for the non-invasive strain, for example, we saw a really high, a high proportion of bacteria that are luminally associated as compared to the wild type. And then you see this clearing. By 6 and 8 hours, the non-invasive strain is basically completely cleared from the lumen. And then you see this decrease as well of the wild type, presumably implicating that it's entering the tissue. And that's exactly what we see when we measure the density. So basically, the density of Shigella within the colonic tissue was calculated over the whole colonic volume that we sampled. So basically, we totaled the count of tissue-associated Shigella and divided that by the total volume of the Swiss roll that we had sampled. So basically, we could calculate that Swiss roll volume because of the thickness of the tissue cut, which was 30 microns. We had a total length of 10 centimeters, and then the width of the approximated width of the mucosa. So this density calculation was really possible because the acquisition protocol we applied, we ensured that really almost all infection foci along that full 10 centimeter length were actually acquired and detected. So as a result, again, we see this clearing of the non-invasive strain, whereas the invasive strain enters the tissue and then continues to replicate over time. And this correlates beautifully with our previous data set of luminal presence of bacteria. So then now we can also examine the depth penetration of Shigella over time. So each individual bacterium's distance was measured from the colonic epithelium, and it's displayed here as violin plots. But it's important to note that these distribution plots are not normalized for bacterial counts. We really just want to look at the collective behavior of the population for each time point, each condition here. So we were able to really measure a well-defined collective wave. So if you really focus on the invasive population, you see this wave of penetration moving deeper and deeper within the tissue over time, whereas the non-invasive strain actually has this retrograde clearing from the tissue. And it's rarely seen, actually, it's also interesting to see that the wild type strain actually never penetrates deep, deep, deep within the mucosa. It never actually makes it to the bottom, which is indicated here. It's actually, you start seeing a retrograde clearing of the bacterium at these later time points. But right now, so far, all the images that we've looked at, all the analysis we've done has been really focused on the bacterial perspective. But we also wanted to measure what's happening on the tissue side. So we aim to robustly measure the area of tissue that was being destroyed by Shigella over time. And this is seen in human shigellosis as well. Shigella literally eats away at the colonic mucosa. So we wanted to measure the volume that is being destroyed. So essentially, we could rely on our manual annotations that we had made previously to use the surface annotation as well as the base to define the mucosal area. And then we defined the lumen, we segmented the luminal space via a simple automatic threshold operation using a percentile method. And then we basically calculated those volumes, the areas of damage, by the intersection of these two masks. So then we have each individual area, but then for the quantification, we discarded really small areas below 100 microns squared because that was mostly due to the normal uninfected surface variability. So we were able to calculate a damage score, which was reported as the sum of all individual areas for each individual field of view that was acquired. And then again, this was per condition per time point. And we see again correlates beautifully with the density of Shigella in the tissue as well as its movement within the tissue, this increase of damage over time for the invasive strain. So essentially, we see that as the damage is occurring, this is correlating beautifully with the bacteria as they're increasing in density and depth penetration within the tissue. So as really a result of all of these measurements, we've gained an insight that we've been able to measure really the dynamics of Shigella infection of the colon. But of course, as scientists, we want more. We want more information so we can actually start to peel away and investigate novel phenotypes regarding Shigella infection. So there had been this growing hypothesis within the lab that Shigella was in fact targeting crypts for early entry into the colonic mucosa. So as I showed you, even at the 2-hour time point, we were seeing this association at these early stages. But we also had observed this in human ex vivo infections at very early time points. Shigella seemed to be hanging around crypts, targeting crypts. So then how can we measure this? How can we see this? So how can we use that based on the annotations we made from this analysis? So in the end, what we did was we focused really on early time points again for the kinetic analysis. This is 2-hour post-challenge time point, because also this time point really ensured the least amount of damage to the tissue in the vicinity of the bacteria. So then it gave us a stronger reliability in crypt mouth annotations, because that was the primary annotation we used. So the distance from each individual bacterium was measured to the closest crypt axis. And this axis was defined as an orthogonal line that passed through the crypt mouth and is orthogonal to the epithelial surface we had delineated previously. So each distance was measured to the closest crypt axis, and then we plotted these distances and generated a radial probability distribution. So if we examine these distribution plots of both populations, the invasive and non-invasive strain, we see that the invasive strain tends towards shorter distances to crypts, well, relative to the non-invasive strain. And this is indicating a bias to these shorter distances, but still we are not sure if this really is a signature of a targeted mechanism. So we need to confirm that this bias is not resulting from random dispersion of bacteria along the surface. So therefore, we numerically simulated a non-biased random distribution along the surface to compare to our experimental measurements. So now we're just looking at the tissue from above. So this is to generate our simulation. So we're looking at the epithelial surface from above, and essentially what we did was we experimentally measured the distances between crypt mouths, and then, which was approximately 70 microns, and then we basically fixed those to a hexagonal grid with that same spacing that we had measured experimentally. And then we randomly distributed bacteria along the surface. And then we generated the same radial distribution plots for this simulation. So again, so here we have the simulation. So the peak is at roughly half the distance between crypts, because then the bacteria will then be associated with the nearest crypt again. But essentially, it followed a very similar, it was more or less a similar spacing to the peak for the non-invasive strain. And when we actually did a comparison, statistical analysis was carried out to really reveal that the actual distances to crypts is not, in fact, random for the invasive strain, and it shows a bias towards these smaller distances, and that, in fact, crypts are being targeted by Shigella. But then, if they are being targeted, what is the target in this case? Are they going in through the lumen? Is there a cellular target at the mouth of crypts that they're finding? So given the bacterial depth penetration, so the main peak of bacteria at 2 hours was approximately 5 to 7 microns from the surface, and what we measured is the proximity to crypts. It's pointing to an intracellular location within the tissue. So it's more or less what we've measured so far is excluding a luminal entry. But now we need to identify those cells that were targeted at these crypt mouths. But in a guinea pig, we really don't have much access to antibodies and things like this. So in the end, we developed a correlative technique. So we used correlative light EM technique, and we applied it to thick tissue sections. So essentially, we had thick colonic tissue sections that we had embedded in agarose and sliced with a vibratome. So we have thick transversal cuts of colonic tissue that we had mounted on Make-Tech dishes, because then we had the grid pattern here. So we imaged them on an upright confocal using a water immersion lens to obtain the XY positions of the infection foci relative to this grid. So we more or less do the XY positions of the bacterial foci of these GFP-expressing Shigella. And then we went on to acquire a higher magnification image where then we could really localize the bacterial foci in Z. So we had the X, Y, Z positions, so we could hand our samples over then to the EM platform. So they weren't just shooting blind, they weren't just slicing for no reason with these thick tissue samples. So we could basically hone in and image again these early infection events. So if we actually zoom in on those images, we could see that Shigella, in fact, were found at the same exact distances that we had measured with the kinetic analysis. And in addition, the Clem technique revealed that Shigella were really found primarily within colonocytes that were really at the mouth of crypts. We could see them associated with crypts due to their proximity to goblet cells. So you can see the goblet cells here. And again, this is another example where you see Shigella primarily within colonocytes. And this is really a novel aspect of Shigella invasion because the paradigm suggests that Shigella actually primarily invade M cells for entry into the mucosa. So this is really just adding a new perspective in Shigella, the studies of Shigella in its host tissue. So basically, just to summarize, we've clearly shown that through application of an organized experimental pipeline and really using really simple analysis techniques, we were able to show in the end that these beautiful images can also provide a great deal of quantitative information. And in our case, we were able to really reveal a novel phenotype specifically regarding our pathogen, Shigella, and that it's targeting colonocytes at the mouths of crypts for early invasion of the mucosa. So again, just stating that this is really adding another dimension to the study of this pathogen's interaction with its host. So then I would just like to thank my collaborators. So this is the laboratory of Philippe. We're here for a reunion in Corsica. And the key players here were friends. Were FX, Julia, Jean-Yves did all the MATLAB scripting and helped us really extract all that data. Of course, Philippe, and then our key collaborators in both the EM and imaging platforms at Institut Pasteur. Thank you.